Hello everyone, I will introduce our virus cleaner service to you, VC for short. It contains three parts: lab situation, VC experiment design, and the virus flow. Our VC labs are constructed with a quality management system based on GLP standards. We also have obtained QP declaration. We have our own cell and virus labs, and both cells and viruses have been backed, so we can conduct a VC independently. Until now, we have 3 VC validation lines that can provide IND and BLA VC services. Here are pictures of our VC equipment and labs. We have 7 protein chromatography instruments and 3 viral retention filtration devices. Virus cleaners can be divided into virus inactivation and virus removal. The steps include chemical methods such as low pH, affinity chromatography, AEX, CEX, and viral retention filtration. These steps can all be independently reproduced by specialists in our VC lab. In addition, based on the different VC characteristics, we will choose the corresponding endpoint detection methods. For example, for chemical methods and VRF, we use infectious methods for detection to exclude the influence of residue nucleic acids. And for affinity chromatography, we will use qPCR method to assess the removal efficiency. Model viruses selection based on the following 3 points. First, relevant viruses identified as potential contaminants or of the same species, such as BVDV, if bomine is used in the process. Second, specific viruses, which are closely related to the known or suspected potential contaminant, such as MULV used in VC of rodent cell production to mimic retrovirus. Finally, non-specific model viruses used to demonstrate that process clearance for viruses is robust. They are selected based on several characteristics, including size, genome type, envelope type, and chemical resistance. Combining regulations and practice, we commonly use typical model viruses such as XMULV, MMV, RU3, and PRV. Our model viruses and indicator cells have been established banned and are all sourced from ATCC. Our viral titers are all around 8 log and will be validated with each batch. IND and BLA have certain differences in their VC steps and model virus selection. We will evaluate this based on your needs and the specific process. In general, we recommend choosing 4 model viruses and doing 2 rounds in 1 batch or 1 round in 3 batches. Our experiments include pre-study and formal study. The pre-study includes 2 parts. First is the cytotoxicity test to assess whether the sample has toxicity to indicator cells. The sample is diluted to varying degrees and added to indicator cells until there is no toxic effect on the cells. The endpoint titration uses the sample at the same dilution. Second is the viral interference assay to confirm whether the sample interferes with the sensitivity of the model virus infection of indicator cells. First, confirm the TCID15 value to the model virus infection of indicator cells, and then add the test article. If the TCID15 value decreases, it indicates that the sample has an inhibitory effect, and the sample needs to be diluted. In the formal study, the endpoint titration is based on TCID15 value. qPCR methods can also be used for quantitative detection of nucleic acid copy numbers. Our VC flowchart is about 6 steps. First, we will provide you with a questionnaire to fill out, which will include some basic information and needs. After evaluation, we will draft a proposal to you. After signing, our project department will communicate with you, including process parameters and sample sending. After that, our operation department will draft a detailed study plan for you to review. Then we will carry out study. Finally, we will draft a study report to you. Above is an introduction to our VC service. Thank you.